Okay, hello everybody and welcome to this new RNA collaborative seminar today. And I'm here as the representative from the French RNA network, which is called the GDR RNA, where we study RNA as a tool and a target for medicinal chemistry and chemical biology. And I am happy today to introduce the first speaker of the session, who is Professor Patrice Gouet from Lyon. So Professor Gouet obtained his PhD working in X-ray crystallography in the University of Paris-Orsay, one of the biggest, maybe the biggest university in the south of Paris, in France, in 1992. And he also did a postdoctoral stay in structural virology at the University of Oxford. He first got a permanent position as a CNRS research scientist from 1998 to 2005, but in 2005 he became professor of structural biology at the University of Lyon. And he is head of a research team specializing in the structure function analysis of viral and ribonucleoprotein complexes. Patrice also develops bioinformatics tools within his teams, and his talk will focus on a new N.ESPript server. And so I'm happy to leave the screen to Patrice for his presentation. You can share your screen whenever you're ready.
So thank you a lot, Maria, for the introduction. Thank you also for the invitation. So I'm about to share my screen. So now it's done. So everything should be okay.
Okay.
So yes, today, so today I'm happy to talk about a new web server we are developing in our team. So this new server is named N.ESPript. And this server, is done for the RNA community. So I will develop things about this server. And the first thing is, if you want to use this server, here you have this link. And at the end of my talk, I will show you how to use this server. So to start with from the beginning, so yes, in the team, we are interested in the development of bioinformatic tools. Okay, and the first tool which was developed in the lab is the program N.ESPript. And the aim of the program is to produce figures with 1D and 2D information, as you can see here. And this is done for publication. As you can see also, my lab was first interested in proteins. So first, we were focusing on proteins. Then we developed a new tool, which is named N.ESPript. And with this new tool, you can also have information on contact between your protein of interest and ligand, which are, of course, bind to this protein. So, for example, here you have a red star, which means that this aspartate binds a calcium ion. Then here we come to one of our last creation, which is the FalseScript web server. And the aim of the server is to compare models predicted with AlphaFold. So for example, here you have two models predicted from the same sequence. You can see that you have different assemblies, and you have also information in color here on the PLDT score of each model. This is a local score, and from this you can have information on the confidence you have on these two models. This screen is just to introduce myself. So yes, I'm professor in Lyon, and I'm the main developer of the core program. But everything is so done with the engineer in our lab. This guy here, Xavier Robert. And Xavier Robert performs the development of all the server I just talked about. So he's performing the development of the interface of N.ESPript, N.ESPript, and FalseScript. As you can see here, these web servers, they are quite well used, through the world, and they are mainly used in the US, in Europe, in China, and in India. But now move to our focus today. So this is this new server, which is named N.ESPript. So it has just been released. Okay, we finished the doc, I mean, this morning. I will show you just a glimpse about the doc. And the aim of N.ESPript is to render sequence and similarity information, and also secondary structure information from aligned sequences. And of course, now we are focusing on nucleic acid because this is a new interest in my team. And at the end, the aim of PolScript, this is for analysis of 1D and 2D information, and of course also for publication purpose, I mean, to make figures. So here I show you a figure which has been made with N.ESPript. So, I mean, what is the input? So here, the input, this is coming, as you can see here, from a multiple sequence alignment. So, for example, here we have an alignment, okay, here, which is coming from crustal omega. And this is an alignment of tRNA sequences, okay? So you use this alignment in N.ESPript and you have this output. But you can also add information on a sequence of interest. So here my sequence of interest is 1EHZ, okay? The first one on the top of this multiple alignment. This sequence has a known structure, and this structure is available in the protein data bank, in the PDB. So you can also upload your PDB ID in N.ESPript, and then information on secondary structure element will be extracted by DSSR. Okay, so our web server is using DSSR, and from DSSR you get this dot bracket notation. So this is what you can see here, and of course I will come back. I mean, what it means. I mean, this information and mainly the color. You have also information here. You can see these gray stars. This means you have modified nucleotides in the PDB files. Now, if I come back to the multiple sequence alignment, I mean, if you have a column of residue with a red background, this means strict identity. If you have residue with a blue frame, I mean, for example, this column, this means that by default, you have more than 70% of strictly conserved residue. I mean, here this is the adenine, which are conserved. And if you have no frames, like here, this means this is remaining. So this is weak conservation. So you have 3 modes in N.ESPript. And the first mode, I mean, the more simple mode, this is the beginner mode. Okay, so you start with the beginner mode when you access to N.ESPript. I will show you that. But you have also access to the advanced mode with more options and the expert mode with even more options. So what you can do if you use the advanced mode, for example, is to add marker. So here, the aligned sequence, they come from a tRNA. And here, just between these 3 residues, you have the anti-codon loop, okay? So you can highlight the anti-codon loop, such as here with red triangle. So this is possible with N.ESPript, and I will show you at the end how to do that. And you can also automatically extract DBN information from all the sequences, assuming that, of course, they have a known structure, and assuming also that you are using the PDB ID. Okay, so here in the sequence alignment, it is extracted from the PDB, and I'm using the PDB IDs in this alignment. So this is an overview on what you can do with N.ESPript and these 2 modes. And now I will get in more details and show you some example. So if you're interested in using this program, you just have to click on this link here, so nespript.ibcp.fr. And if you do that, you will have access to this form. And on this form, you start with the beginner mode, okay? So this is this red button, big. So you start with the beginner mode. As I told you, the main information you will give to N.ESPript, this is a multiple sequence alignment file, okay? So here this is an alignment from tRNA sequences, and they are all of known structure. And first, for my example, I will focus on the first sequence of the alignment, and the first sequence of the alignment is the PDB ID 1EHZ. Okay, so what you just have to do is to upload your multiple sequence alignment files, for example, coming from Clustal Omega. Then you upload your PDB file. Okay, so now it's done. Once it's done, you go up, you press on the submit button. Okay, done. And after a few seconds, you will get the result button. Okay. So from this result button, you can have access to other information. So you can check here the log file if you want more detailed information. And you have the result. And the result, this is a PostScript file. I mean, this is why it is named N.ESPript. At the end, IPT, this is for PostScript file. So you get this PostScript file. This is just what I showed you on my previous slide. And you have information here on the conservation, on your alignment, in quite nice color. And also, I mean, with color, you have information on secondary structure elements. Okay. So now I will go again in more details on the information you obtain with N.ESPript. So just what I have obtained, this is this figure here, okay? So now if I focus here on the DBN notation, so in blue bracket, you have regular bases, base pairs. If it is orange, like between this nucleotide and this nucleotide, this means you have a wobble pair. If it is a bracket in maroon, this means a pseudonodes, for example, as between this nucleotide and this nucleotide. And if you add more information, I mean, if you add, for example, a Varna figure, you will maybe better understand the information now I'm talking about. So here, this is a tRNA, and on this tRNA, you have 1, 2, 3, 4-way junctions. So you can see that on the N.ESPript figure, P1, P1, and P1. Okay, so you know exactly the sequence here, and you know the sequence on the alignment. Here you have 3 hairpin loops, okay, eta 1, eta 2, eta 3, eta 1, eta 2, eta 3. And at the end, you have a non-loop single strand, okay, which is named alpha 1, okay? So you can compare information here on the MSA and in Varna. And at the end on eta 2, you have the anti-codon loop. And here you have the final 3D structure figure made with Chimerex. And the I-time codon loop is just right there. So here you have a global view of what you can do in the beginner mode with N.ESPript. And now I will move to a bit more complicated figure, okay, that you can see here. So now on this figure, I will highlight the third sequence, and my reference PDB will be on this sequence, okay? The third one on the alignment, which is named 6AH3, and I'm looking for the chain T of this PDB file. And also, you will see, I just have to press one button, and you will have the DBN notation for most of the sequence, okay? So now, let's move on and have a look on N.ESPript. So I click here and doing that online. So now my browser is open, okay, linked to N.ESPript. Here, this is the first page you obtain, okay, so here you have an example figure, and this is performed from the alignment I'm talking about. You have access, so we just finished this, this morning, to full documentation, okay, which is here. You have access also to a tutorial. So, I mean, if people are interested, you really have to do this tutorial. You have a few examples, 1, 2, 3, okay. And with this example, I think you can really understand how to work with N.ESPript. We have a gallery, and we have how to cite N.ESPript if you're interested to use it for a figure. So now I will run the program. So you press just here, run N.ESPript, okay. You have access to the form I just showed you. By default, this is made for RNA sequence, as you can see here. So now I will upload my alignment. Okay, so the button was in French. I upload my alignment made with Clustal. Okay, done. I upload my PDB file. So my PDB file is the one of the third sequence in the alignment, which is named 6AH3. And I'm looking, okay, on the chain T, which is the tRNA. Okay, done. And now I will just, okay, so the 70% score is coming from this button. This is the default, so this is the blue frame. I will use all the sequence in the alignment. You can play with the layout, not going to play with that. By default, you get a PDF file, but you can also obtain images. So now I go back, just submit. Okay, you can see you get an immediate result. And you have this result, okay? So as you can see here, N.ESPript is naming this DBN 1EHZ, because by default, it thinks this is the first sequence, okay, 1EHZ. But in this case, this is not true. I'm referring to the third sequence, which is named 6H3 underscore T. So I will correct this. And the second information now I will enter. This is the anti-codon loop, which is just here. So I go back to N.ESPript, okay, which is not this one. This is this one. And now I will move to the advanced form by clicking here. So you can see you have more options. And if I go here, I can specify which sequence my DBM is assigned to. So it will be assigned to the third sequence. So the command is XB for blue 3. So you will see the name is in blue. Now I just add that I want also the title of the sequence, T in the alignment in blue. And I want also the numbering to be referred to the third sequence. I will also check this button, which means you will see, you will distinguish between purine and pyrimidine residue. It's red and blue. You will see that. And to finish with, I will enable this button, meaning I want to display all the structure from this alignment, okay? And this will be performed because all these names, they are coming from the PDB. Now I press the submit button. Sorry for that. I made an error. Wait, correct. So you see, you can make error. It was B, why B for blue color. Made this error. Submit again. Here we go. No error this time. PDF. So now no problem. I'm referring to the third sequence, which is okay. Here you see all the DBM which are on displayed. And you see also that you have black dots. So these dots means, for example, if I consider this structure, this is a low resolution structure obtained by cryo-EM, okay? And since this is a low resolution structure, in this structure, you only have the P trace, only the phosphorus of your nucleic acids. So DSSR will not be able to extract secondary structure information, okay? So that's why, I mean, you keep these black dots. So I go back to N.ESPript, and just to finish with, you can see that the anti-codon loop, it's from residue 39 to residue 41. So go back to N.ESPript, just have a problem with, here we go. So now I will say that I want to highlight my anti-codon loop in red, okay, with a down triangle, okay? So this is this D command. So I say D, not U, which is up, down triangle in red for residue 39 to 41. Then I say, okay, I want to get rid of the low resolution structure, just have to press on this button. And to finish with, I have exactly the same thing as in my figure. I will say that I will just select sequence number 1 to number 9, and that's it. I click on the submit button, and I have, in a few minutes, my final figure, as you can see here. Okay, so and you can distinguish between purine and pyrimidine, between the red and the blue. So that's it for the demo. Come back to here. Okay, this is how you can obtain this figure in just a few minutes. I thank you for your attention, and I'm ready to take questions.
Okay, thank you very much for this excellent presentation. Let me check if there are already some questions. For the moment, I don't see them, so I would like to tell to the participants to not hesitate to put your questions on the chat, the question and answer window, actually. So maybe I can begin with a short question, I guess a naive question.
Sure, Mario.
So you start with a PBD.
Yeah.
Structure, right? What happens if you don't have the PBD structure?
You can upload it. If you go back, so yes, I mean, I think that most of the questions that will be on the web server, which is new.
Sorry.
So yeah, so thanks for the question. I just, okay, I just have little problem. So yes, you can choose to upload your own PDB file. I'm just trying to show you.
But you need a PBD file in
Of course,
first
okay,
place.
you need a PDB file.
The 2D structure, I was wondering if the 2D
structure
is predicted by the system or not.
Okay, get it. Okay, so we will answer with the form in background. Okay, so there is two answers. So the first answer, if I click on the PDB logo, you upload a PDB ID. Okay,
yeah.
So currently in the PDB, you have about 250,000 experimental file structure, which have been determined by either X-ray crystallography, NMR, or cryo-EM. You can upload that. And in fact, we have this data in our own server. So in fact, you are uploading the data from our own server. But you have also access to models. There are about 1.5 million models which have been predicted by AlphaFold. And we have them also on our database. So it is possible to use our database to upload this model. So this is the first answer. The second answer is if you click here, okay, on this button, you can upload your own model. So it can be experimental or it can be your own. So you can upload a file. And just for information, you can upload a PDB or you can upload a DSSR. I mean, they are both supported by the program. And just to finish, as you've seen, I mean, this is a 0.9 version, okay? It's very new. So if you have suggestion of if you see any bug, we will be happy, I mean, to fix them and to improve the program. So this is really a first version, and we are happy to show this version to the RNA community.
Okay, thanks. Good. So let's see. There is a question now in the chat. So the question is, do I have to provide the 2D structure information in the MSA file?
No, in the MEC file, I mean, it's only, it is an MSA file. So this is just your alignment, okay? From Clustal, Omega, Multalin. I mean, most of the formats are supported because here, in fact, this is the same format as the form we are using for NSPript, okay? And we are using this form, I mean, it was created about 20 years ago. So just the answer is no information about the secondary structure element on this section, and structure information is on this section, okay? But there is a but. What you can do, just for information, you can also, I mean, it is, and I will finish, sorry, I still have 5 minutes. So just what you can do also, you can upload a single PDB file if you're interested. Okay, so I will show you that with this one JJD, because I mean it is exemplified. So I just retrieve data. Okay, so now I'm using it for the alignment. I'm using it again, okay, as you can see here, to extract secondary structure information. So you have to upload here and here. And now I have to specify that I'm looking to chain ID 9 here and to chain ID 9 here. Okay, so this is your RNA. You just press submit and you obtain this directly. Sorry. Up. Yeah. Okay. So this is extracted from the first section, and this is extracted from the second section. So you can upload also PDB information and have directly the DBN information from that. So this is possible as well.
Okay.
So not only MSC, PDB as well.
Thanks. Another question, can I select the multi-chains at the same time, since some RNA has interactions between different chains?
Not well, I'm not an RNA specialist. Sorry, to answer. Okay, as you see, you have to select one chain ID. Okay, this is the first thing. So anyway, you have to select one chain ID. Okay, now the second thing, actually, it is not possible with N.ESPript to display contact. It is not possible for the moment. But as I said, we have another web tool, which is named N.ESPript. This is for proteins. And with this web server, you can display protein ligand contacts. So next year, we will upgrade this program, and for sure, we will add this tool. So it will be possible as well, if you upload a PDB file, to have also information, if it is crystal, between nucleotide and also between a nucleotide and ion or ligand, etc. Okay, so yes, we will do that, but next year, okay, because we have to upgrade the program. So it is not possible for the moment, but it is planned for sure. Yeah, so that will be possible.
Okay, thanks. I think we can close this first part of the seminar. Thank you again for the presentation.
Thank you, Marianne. Thanks everyone. So I just
and
stop sharing.
leave the screen to our colleagues from China. So you're welcome to begin.
Thank you. It's a great honor to do together with colleagues in RNA folks in France. So now it's my turn. So I'm Xin Li I'm the center director of the University of the Zhejiang University's Center for RNA Medicine. Today, our speaker is a graduate student. Huisen Zhan is actually the first time our center has student presenter. Mr. Zhang got his undergrad and in Zhejiang University, and now is the final year of his PhD. So his mentor is Dr. Mingxin Guan, and his presentation is on the tRNA in the mitochondria. So Huisen, now it's your turn. Please go.
Okay. Thank you for your invitation, Dr. Li Xin. And today I'm going to share my a part of my work during my PhD student time. And my title is about the MELAS-associated tRNALeu UR3243A2 gene mutation, how it reshapes the mitochondrial integrity with distinct fission signatures. And I will introduce some basic backgrounds about the mitochondria and the tRNA things at first. So the mitochondria has its own genome, and basically the human mitochondria encodes 37 genes with 2 RNA and 22 tRNA and 13 mRNA. Besides these 37 genes, there are about 14 100 proteins were encoded by the nuclear genes. So basically 99% of mitochondrial genes are coded by the nuclear genes. And for some backgrounds about the pathogenic mtDNA mutations. First, because the mitochondria in a single cell is displayed with multiple copies, so it can vary from thousands of copies to hundreds of thousands of copies. And in this situation, if someone carries mutant mtDNA, and it displayed with a certain proportion, varies from 0 to 100%, and it can be verified. So it was called the heteroplasmic mutation. And if someone carries mutant mtDNA with in his cells, there are all of the mtDNA are mutant, so we call it homoplasmic mutant mtDNA. And because during the fertilization, the oocytes will preserve the mtDNA, while the sperm will lost all of these mtDNA. So the inheritance of the pathogenic mtDNA also displayed with maternal inheritance pattern. And also in the development of the embryonic, in the embryonic development, you can see because the eggs after they divided, and the nuclear DNA will produce will replicate it and divide into each cell. But since the mtDNA in the oocytes keeps the same number, and it don't have enough time to replicate compared to the nuclear DNA. So if heteroplasmic pathogenic mtDNA mutation in the oocytes will distribute it in the blastocytes with a different distribution. So it can vary from 0%. So it basically means if someone's mother has a certain ratio of mutant mtDNA, it can vary. His children can vary. Her children can vary from different level of the mutant loading. And after that, I will introduce the one of the most common pathogenic mtDNA mutation, which is the MELAS-associated mitochondrial tRNALeu UUR 3243A2G mutation. The MELAS, its full name is mitochondrial myopathy encephalopathy with lactic acidosis and straw-like episodes. Its clinical manifestation include the straw-like episodes, brain damage, the diabetes, and people can find elevated lactate in its blood and the cerebrospinal fluid. And also, most of the MELAS patients, they behaved with hearing impairment. And this mutation, the 3243A2G mutation, is spread in the population with about 200 in each 100 and 1,000. And there are multiple research has shown that in the diabetes patients in different cohorts, there are about 2% are carriers of this mutation. The 3243A2G mutation is first discovered in the 1990 by the Gouet. And after that, people have researchers have do some investigation about how the mutation affects the tRNA metabolism, and after that influence the mtDNA encoded protein synthesis. So basically, at first, people found it will influence the tRNA structure and the stability. And also it will influence the post-transcription processing because the mtDNA is double-strand circle. And at first, it's long transcripts, and it requires post-transcription cleavage, and after that, you can get the mature tRNA or the RNA or the mRNA, and also the mutation will influence the amino acylation, so it's basically influence the charging of the tRNA, and after that. People also find it will influence the bubble base pairs modification. And finally, it will influence the mtDNA-encoded protein synthesis. And beyond these basic concepts, people also find there is the mutation happens in the mTORF binding sites. So the mTORF is the transcription factor of the mitochondrial DNA transcription, and it will influence the mitochondrial transcription and DNA replication. So it has a multiple impact of to the mitochondrial activity. And to investigate the cellular effects of this mtDNA mutation, we are trying to keep the same nuclear genome across different cell lines. So basically, we use the 143B derived cells, Rosero cells. The 143B is a thymine kinase carries with thymine kinase deficiency. So basically, it can be resistant for the BRDO treatment. And the Rosero cells, it comes from the 143B with treatment with the estimating bromide. And after that, the replication of the mtDNA is blocked. And with a long-term treatment, the 143B will come to be the Rosero cells. The Rosero cells carries disappeared mtDNA. So basically, it's mtDNA-less cells, and because the mitochondrial function was disappeared, so the Rosero cells carries the property of DHODH enzyme deficiency, so it requires extra uridine to make it can grow normally. And in this way, we take the myoblast from the patients and extracts its cytoplasts. And after that, fusion with the Rosero cells. And we selected with the media with BRDO treatments and don't provide extra uridine. So after the selection, we can find the cyprates with the same genome background, same nuclear genome background, which is from the 143B nucleus, and also you can find the mitochondria from the donor cells, which is the patient's mitochondria, will replace the mtDNA-less mitochondria in the Rosero cells. And since we are investigate a system with mitochondrial dysfunction, so it's I need to talk about the basic concepts for what happens when there is something wrong goes in the mitochondria. So in the protein level, there is a UPRMT signaling pathway to regulate the unfolded proteins in the mitochondrial matrix. It will activate certain chaperones and proteases. And if doesn't work goes well, and it will start the octanol level system to control the mitochondrial quality. And in this way, the most usual we talk about is how to rebalance the mitochondrial dynamics and use the mitophage to remove this part of damaging mitochondria. And also there are some unusual way, like the mitochondria-derived radicals or the exocytosis on the mitochondria is some also plays some roles in the control system on the mitochondria. So at first we try to investigate whether this mTDA mutation will affect the OX4 systems. So we use the blue native gels to isolate different complexes and try to find what's the problem inside the OX4 systems, and then do the in-gel activity to check the enzymatic catalysis in different complexes. So basically, we found both the complex 1, complex 3, and complex 4 and complex 5 were impaired. In these complexes, the complex 3 has returned for its individual part, but it cannot assemble with the complex 1 or complex 4. Obviously, the complex 1 and the complex 4 are very sick, so they cannot accompany together. And the complex 2, because it's the only one complex that all of its components are encoded by the nuclear, so it's affected very small, very mildly. And also the catalytic enzymes, their activities were checked, and the complex 1, complex 4, and complex 5 almost lost their catalytic activities are almost lost. And the complex 2 keeps a weaker enzyme activity. I think it's because it's a whole system. So the loss of the certain enzymes will influence the whole mitochondrial membrane potential and make it difficult to synthesize the component of the complex 2. And after that, we try to figure out what the cellular effects to influence the mitochondrial activity. So first, we see the morphology of different kinds of cells, and here we find the mitochondrial morphology in the mutant cells was significantly changed. It displaced with the smaller and rounded mitochondria. And also we found that in the transmission electron microscopy, to make sure the mitofission is on the way to control this site. We try to use the immunofluorescence to label the cofactor to regulate the mitofissions part. So you can see I use the mCherry DRP1 to locate the mitochondrial fission events, and the mutant cells displayed with high frequency of the mutant of the mitochondrial fission. And we want to find what's the major factor to drive the mitochondrial fission in the mutant cells. Obviously, the cellular effects is to separate this damaged part and find some way to dispose these part. But we don't know what's the major factor to drive this way. And we checked the most of the mitochondrial dynamic related proteins and find only the fifth one is the major factor to drive this process. However, the major way of the mitochondrial fission in a complicated field today, basically people think there are two major ways to drive the mitochondrial fission. One is the mid-zone fission governed by the MFF, and it leads to the proliferation of the mitochondrial population. And another one is the peripheral fission. It drives by the fifth one. The purpose of the peripheral fission is to separate the damaged part and dispose it. So to separate this part, we need to find whether the fifth one and lysosome in this way, the lysosome contact dependent this peripheral fission happens in the mutant cells more frequently. And there is another problem is that although the role of the MFF is very clear, if people knock down the MFF, and you will find the network of the mitochondria are elongated. However, if people knock down the FIS1 in multiple mammalian cells, it didn't affect the mitochondrial morphology. So it remains elusive. The human FIS1 can play a role to drive the mitochondrial fission. So we take a glance how it happens in the purification, and we use the lyso checker to label the lysosome and mitoGFP to label the mitochondria. So we found in the mutant cells, the 43B, the mitochondrial lysosome contacts mediates the purification. You can see smaller daughter mitochondria are divided, and the mitochondrial lysosome contacts in the mutant 3243B cells, its dual rate longer and with displayed with higher fission frequencies. So basically, we can say the purification is promoted, but is the FIS1 or REP7A do play their role in the purification? So we knock down the FIS1 and the REP7A in both wild type and the mutant cells. And interesting, we found if you knock down the FIS1, it will induce the reduction of the REP7A, and vice versa. If you knock down the REP7A, it will induce the reduction of FIS1. It implies there must be some public regulator upstream of the FIS1 and REP7A to regulate the purification. But then we first to take the to examine whether the knockdown of the FIS1 or REP7A will affect the mitochondrial morphology. Obviously, knockdown of the FIS1 or REP7A reduced the piecemeal mitochondria in the mutant cells. However, it didn't have an impact for mitochondrial morphology in the control cells. So it implies there is a selectivity. To examine whether these separated damaged mitochondria will be removed or some way. So we check the autophagy things to make sure that the destiny of these damaged mitochondria. The macroautophagy or the autophagy is a way to engulf different unused stuff or damaged organelle into a membrane and fused with the lysosome to degrade these things. So we first use the dual fluorescence of the tagged with the LC3. It's a marker of the autophagism. And since the EGFP is easy to quench in the acidic pH in acidic environments, so basically the red punctate means the autolysosomes, and the green or yellow ones are the autophagisms. We found that the autophagisms accumulates in the mutant 3243 cells, but there is a problem. Since the autophagism, in the one side, the autophagisms are produced, and in the other side, these autophagisms are also be reduced or recycled with the lysosome. So we need to block the lysosome with the buffermycin and to check the flux during the 24 hours. So we also found that the LC3 in the 24 hours are accumulating more in the mutant cells. And also we found some autophage structure in the transmission electron microscopy. So we can say the macroautophagy in the mutant cells are promoted. But we still need to clarify whether the cargo in the autophagy are those damaged mitos. Similarly, we use the dual fluorescence of two targets, the mitochondrial outer membrane, and we also find the mitophagy in the mutant cells were intensively induced. And to make sure the selected the certain signaling pathway take part in the mitophage. In the mutant cells, we check different, both the pink one packing dependent mitophage and the mitophage receptor. And we found at least the pink one packing dependent mitophage is promoted in the mutant 3243 cells, but some mitophage receptor in the mutant cells are reduced. It might be degraded during the mitophage. It also can be because they are downregulated, so it's still elusive. And also, we concerned whether the fission and the rep7a is essential for the process of the mitophage in the mutant cells, because since it plays a role in the separate different damaged metals. So the damaged metals need to be to be dealt with certain way to remove. And in the knockdown cells, we found both the macroautophage or the mitophage pulpita are reduced after we knock down the fission or rep7a. So it means the fission and rep7a is not only play a role in the perforation, it also needed for the mitophage. As I have mentioned it before, the fission and rep7a has a similar expression pattern, very close expression pattern. So we assume there is a potential regulator of the fission and rep7a. And this regulator, it must take part into the mitochondrial division and also the autophagy or degradation of damaged organelles. So we try several signaling pathway and found is the AMPK signalings. We found the AMPK is activated in the mutant cells, while it's also the activity AMPK will inhibit the mTOR signalings. And if we use the drugs to inhibit the AMPK signalings, we can find both autophagy and the perforation related proteins are downregulated after the AMPK inhibition. And also we can find with the AMPK inhibition, the fragmented mitochondria were decreased in the mutant cells. However, as I said, because the AMPK signaling only activated in the mutant cells, it influenced only the AMPK inhibition. It also only influenced the mutant cells. And AMPK inhibition also will impede the mitophage in the mutant 3243 cells. To be summary, in the healthy mitochondria, there is equilibrium of both degradation and biogenesis. So basically, FIS1 and MFF has placed their role. And in these mutant 3243 A2G mutation cells, the AMPK FIS1 rep7a axis lead to the asymmetric fission and mitophage. In this way, it can restrain the ROS generation within autophagons and won't affect other compartments. However, the overactivated mitofission and mitophage will cause the leakage of the mitochondrial matrix contents like cytochrome C or calcium, which will induce the apoptosis. And also, it's a persistent of mitophage and mitochondrial division. It will waste the energy. However, there is another situation which I don't mention here, but is also part of my job, a part of my work. There is the MELAS-associated 8344 A2G mutation. In this cell, it has a severe mitochondrial dysfunction, but the AMPK signaling is inhibited, and the lysosome function is suppressed, and it will induce the mitochondrial secretion. So basically, the mitochondria are not degraded intracellularly. It was delivered to the outside. And in this way, it will save the energy, and the content in the mitochondrial matrix will not enter the cytosol, so it's more safe. But the problem is it will induce a high level. It will keep a high level of ROS, so it will generate oxidative stress environments for other organelles. And there was some thinking about the different tRNA mutation cause different problem in the cells. So first is the crosstalk between the mitochondria and lysosomes. We have shown that the lysosome contact is very useful for clearing the damaged mitochondria. But how this happens, where is the information starts from the rows from some direct contact, because there is some research have shown that the mitochondria and lysosome can deliver some ions as the information. And also, is the peripheral fission a prerequisite of the macrophage? Because the FIS1 and the RAF7A is important for the macrophage, and the accumulation of these damaged mitochondria, they need to separate this damaged part, but we still need to propose this substrate of the macrophage, right? And finally, why the AMPK plays a role to regulate both the mitochondrial damage and, in fact, because the AMPK is rapid responsive kinase to rescue the lacking to rescue the lacking of the energy. So if a persistent activation of AMPK, it might be detrimental for the cell survival. So different strategy for different tRNA mutations. I think that's all my speech. Thank you.
Okay, thank you, Huisen, for this very combine the biochemistry and the physiological of the mitochondria, like into the RNA, extended RNA's the study. So I would like to begin. So you mentioned like this mutation is high in diabetes patients. So which type of diabetes?
Type 2.
Type 2. Okay.
Yeah.
So it's more frequent. And is that other tRNA, because mitochondria has multiple tRNA genes, so has that been found, the similar phenotype been found in other tRNA, like
yeah, I have to say, I think it's multiple aspects to describe this question, because first, the most severe tRNA mutations happens in the heteroplasmic mutation. So like the 3243Hg mutation, when it's in a low ratios, like 20 or 30%, it can induce the MIDD, which is the mitochondrial diabetes with the hearing impairment. And for some other kinds of homoplasmic mutations, it can be maybe, only influence the hearing, even it's 100%.
What I mean, affecting the mitochondria, like the fission, that other tRNA mutation also cause that?
Not always. So
No.
some tRNA will influence the mitochondrial activity, like the mitofission or the mitophage. Some others won't. Yeah, and because the remaining genes were encoded by the nuclear genes, there are also some research has shown that if you knock down or knock out some mitochondrial genes, it has displayed with different kinds of situation. Some will influence the mitofission and mitophage, some others don't. Yeah.
So do you think that the difference is due to translation of in the mitochondria, or is it due to another outside the tRNA function outside of the translation?
Because I spend most time in the tRNA-leucine and the tRNA-lysine. So I think most in the mitochondrial damage, they displayed very similar influence, but in the cellular effects, it's totally different. And so in this way, I should say the protein translation, yes, they have several different, they have some differences, but it's not enough to induce the different cellular effects. But because I have mentioned it, there is mTOR binding sites in the 3243A2G mutation. So it's not only a problem of the translation, it might be a more complicated question, like the transcription and the replication of the mtDNA. So I think it's more than that, not only a problem of the translation, of the protein translation.
So since this mutation, I didn't see other questions, so I would continue to ask. So since this mutation only happened in the, so it's very frequent. So I wonder whether it's always de novo derived, or it's that it's existing in the population, like very, very low percentage, and then in some patients it's become more expanded.
I think both happens in the cohort, and I think because the heteroplasmic can distribute it in the childhood, in the children with different ratios. So most are still not the de novo mutations, but it also happens.
Okay, I think we have to, the time is up, so we have to conclude the section. So thanks, Huisen Zhan, and thanks for the previous speaker. Okay,
Thank you.
okay,
Bye.
bye.
Bye.